So let me know if you're ready. We can see your screen now. Thank you.
Yes, please let me know when to navigate.
Thank you so much. So I'll put on my screen real quick, so hopefully it works. Okay, so this work, as you know, has been done in collaboration with the Brighton Collaboration Group, SPEAC, and also supported by the CEPI through SPEAC. The next slide, please. So this is just to give you an idea. Many of you have participated in Brighton projects, and you can see the process here was relatively compressed in that early in March when the group was called, and after participating in an important consensus meeting looking mostly at the preclinical data available for vaccine-associated enhanced disease, the group got together to define the scope of the work, have an outline of the manuscript that we would be putting together, and as usual, conducted literature reviews to be able to participate with the different topics assigned to the members. And we were working throughout most of the spring and summertime in completing a manuscript that was then submitted to peer review. This manuscript review through the Brighton collaboration process received many reviews. So if you were one of them, thank you for that. And we also had expert reviews, as you can see, dedicated to looking at the document in detail. We are at the final stretch and expect to have this manuscript ready for submission in the next few days. I have to say, having put together several case definitions, certainly the vaccine-associated enhanced disease has been relatively challenging, not just because of the timeline and the need to have this ready for vaccine development for COVID-19, but also because of the topic, and I will try to share some of those challenges with you. The next slide, please. The working group members were selected based on their expertise. As you can see, many of the work group members have lifelong careers in the area of vaccine development and also the assessment of vaccine-associated enhanced disease. And as you can see, we had also broad geographic and expertise representation. And so everyone contributed greatly to this work. Next slide, please. So again, I'm sure most of you are familiar with the process. Just to briefly recapitulate what the Brighton process is. Remember that the purpose is to have a consensus case definition to assess safety of vaccines in the context of vaccine development. So most case definitions are geared towards the assessment prospectively in the context of a clinical trial. But we do anticipate that the case definitions are broad enough that they will be utilized in all cycles or the life cycle of the vaccine, and potentially be also utilized in epidemiologic studies, observational studies. The goal of the case definition is to standardize a particular term, not to assess causality. That needs to be clear. And with the standardization, we seek to have consistency in the assessment of the event, harmonization in the way that the event is evaluated, and that allows for comparability between different studies and different reports and publications. And the basic format is, as you know, usually 3 levels of certainty that are key, where the first level is have the highest probability of having a case, definitive case, and that would be considered the gold standard for a case definition. And you would have, at that point, access to all the diagnostic testing that you need. While levels 2 and 3 will give you a probable or possible case with a lower level of certainty, but certainly still good sense that this could be a case of the topic decided. Next slide, please. So in this case, thank you. In this case, vaccine-associated enhanced disease. This is a general term. There are other terms that are utilized in the same way: vaccine-mediated enhanced disease. This is a phenomenon that had been described already with other vaccines in the past, for example, RSV, measles, some influenza vaccines. And the general concept is that this is an event that may occur in people who receive a vaccine, and then they are subsequently infected with the pathogen that the vaccine is meant to protect against. And they do have, at some point, either a severe disease or a different modified or unusual presentation of the disease. So as the beginning point, this definition requires a clear understanding of the natural disease to be able to compare, and it's a relative evaluation to that natural disease to know that you do have that enhancement or that more severe presentation or modified unusual presentation of that known disease. So it's a relative term. It also is usually known to be associated with respiratory illness. So we talk about vaccine-associated respiratory disease for the most part with some of those respiratory virus vaccines. But we also know that, especially in the case of COVID, it can involve multiple organ systems. And at this point, though, although the definition and the development of this definition was inspired by COVID-19, it's still a theoretical concern as to whether vaccine-associated enhanced disease could occur or not with COVID-19 vaccines. I think that is important to know. So again, the scope of the work was to define vaccine-associated enhanced disease in general, and then did also mention vaccine-associated enhanced respiratory disease to focus on the lung. The approach was for clinical trials to look at clinical presentation complemented by epidemiology and also suggested laboratory evaluation, while for case identification post-implementation or observational epidemiological studies, definitely more challenges to be able to ascertain, and this is why we would like to have your input as well in this conversation. The next slide. So much has been published in the context of COVID-19 regarding vaccine-associated enhanced disease. I think most of you must be familiar with Barney Graham's recent publication. And I like this table because it really identifies the key pathways to vaccine-associated enhanced disease. So we put them in two categories. One would be an antibody-mediated pathway, and the other a T-cell mediated pathway. You see how the T-cell is where we look at a situation where you have a Th2 biased immune response with an exaggerated or exuberant inflammatory response, usually seen through the assessment of Th2 cytokines, but also a differentiation where you do have biased CD8, CD4 balance. And this is what we usually see with respiratory disease in the past with RSV, for example. Antibody-mediated, two types. You could have that respiratory disease, where in this case, we're talking about immune complex formation, complement deposition and activation, which then results in that inflammatory response, a little bit different. Or when you have the classic antibody-dependent enhancement as seen in, for example, dengue disease, where you do have, in this case, poorly neutralizing antibodies and potentially, again, an increase in viral entry, macrophage activation, and then that development of inflammatory cytokines. So in this context, in general, it has to do with the need for making sure that antibodies are neutralizing enough. So these mechanisms are what has been described. It is important to know that there is no single laboratory test or no single evaluation that will give you a clear diagnosis of any of these components. Next slide, please. So with that in mind, the case definition here is the general set of elements that we have included. And again, we include general systemic enhanced disease and also respiratory disease. And the overall point is that it may occur in a previously naive person who's vaccinated and then is infected with the pathogen targeted by the vaccine. So it's general, it's not to be specific to COVID-19. In this case, the elements include confirmation of the infection with the disease manifestations that we know are related to that particular infection in one or more organ systems. The second element is that relative increased severity of the disease to what is known of the natural disease. And this is where the validity or the importance of having a valid clinical severity illness scores is important to be able to harmonize the assessment of that severity. The next element is a frequency that could be higher than expected. And we'll talk more about this, I'm sure, today, but this was considered important by the working group because, again, at this point, this is one of the ways to identify vaccine-enhanced disease. And we'll talk about the triggers in a moment. But in this case, you would usually need to have either in a clinical setting, a control group, or a good sense of background rates of certain instances of those manifestations. And then the fourth element, I'm sorry, would be the presence of immunopathology in the organs that are involved. And in this case, specifically about histopathology with some supporting laboratory evidence. And importantly, no identified alternative etiology. The next slide will show you just again, the thinking is that you would need to identify triggers to determine whether you are having that increased severity. And certainly either a more severe infection, which could be evaluated with existing clinical scores. I have listed two of them there. For example, the MUSE or PEWS for pediatric scoring system, which is used for outpatient ambulatory patients mostly. And then critical illness scores, such as the SOFA score. There are many of them validated as well. And as well, respiratory illness scores for ARDS, for example, that could be utilized. And I think this is important to make sure that one is selected across the board in a particular study. But you could also have triggers such as increased hospitalization, ICU admission, or mortality that would help you decide whether there could be a signal there. The next slide is just some of the challenges. And this is, again, important in the context of this discussion today when we discuss looking at vaccine-associated enhanced disease in epidemiologic studies. Because when you do have a clinical trial and you have comparison groups that are relatively even and selected, you do have an advantage in that you can assess the differences by breaking the blind. But when you're asking about background rates in epidemiologic studies, there are many factors to consider. Clearly, in thinking again about COVID-19, we know that a particular disease could have different manifestations or severity expressions at different age times. So that is important to consider based on age, for example. We know that the circulation of the pathogen can vary. And so depending on whether it's seasonal or sporadic or even endemic, you may have different time points. And so at what time point do you consider to be the reference for those background rates? Similarly, the different population-specific variabilities that may occur. How do you differentiate this from natural disease or natural infection? The clinical manifestations would be very similar, but you do need to think about then the clinical course and the progression of symptoms. Is it going to be what you expect? And for that, you need to have a clear understanding of the disease itself, its outcomes, expected outcomes, what are the progression or resolution, what are sequelae, what would be complications, etc. And of course, many other confounders that would be other infections, comorbidities, drug effects, other toxicities, just to keep that in mind. Just again, pointing out the challenges. The next slide is just some guidance that was provided by the working group, and this is in the publication and the case definition. without being prescriptive about what types of tests need to be done, because this could be variable depending on the disease. These are some of the diagnostic tools or assessment elements for vaccine-associated enhanced disease. So one would be able to show that there is inadequate or imbalanced neutralizing antibody responses by doing this type of testing. So binding and neutralizing antibodies, the ratios, when you have inability of the antibodies to work, evidenced by high viral load, for example. So those would be, again, suggested evaluations. The other element is here where you have a inadequate or biased cellular immune responses. And in this case, you see how when you have low CD8, high CD4 unbalanced, or lymphopenia, lymphocytosis, or different cytokines that are Th2, that could be consistent with vaccine-associated enhanced disease. And the next slide continues with additional diagnostic assessments where you could have evidence of exuberant inflammatory responses by measuring specific cytokines that are pro-inflammatory, as listed here, or indirectly, a little bit less specific, but other inflammatory markers or biomarkers that you see, such as C-reactive protein, D-dimers, etc. And finally, what was considered to be more a stronger evidence by the group was immunopathology in the organs. So if you have histopathology, we're not saying you have to have histopathology, but if there is the option to have histopathology available, and indeed one of these findings is present, none of them is pathognomonic or typical, but a combination of these with, again, tissue evidence of increased inflammation and injury, that could be more consistent with vaccine-associated enhanced disease in the proper context. The next slide. It's just a final point, to show you the deliberations from the group. Certainly, another set of challenges include the context in which you assess vaccine-enhanced disease. If it's related to a vaccination or temporally associated to a vaccination or not, and the main discussion point has been differentiating vaccine failure versus vaccine-associated enhanced disease. And I think this is a key point because we know that vaccine-enhanced disease could be inherently a vaccine failure. It means that the person who was vaccinated still became infected and developed that enhanced disease. But not all vaccine failures obviously are vaccine-associated enhanced disease, because you could have breakthrough infections, different levels of protection, and so on. So that discussion, I think, is critical, and that recognition that there has to be some at least temporal association with a vaccination and then an infection. But what is that time is really unclear. What is the time of onset after the vaccination or after the new infection that one would consider relative, and important to consider vaccine-associated enhanced disease? And that brings up the question of how long do you follow these subjects or patients after vaccination to try to ascertain this problem? And the solution for that has been varied depending on, different recommendations from regulatory agencies. But certainly at this point, one of the key messages is also to better understand this disease and its manifestations. The next slide is just a conclusion slide to pass it on to my colleagues. But again, to make the point that the Brighton case definition is meant to provide guidance for the assessment of the vaccine-associated enhanced disease for any vaccine, not just COVID. But clearly we are concerned about COVID, mainly in the context of theoretical considerations, not anything that has been proven to date. We know that the potential for or the risk for vaccine-associated enhanced disease for COVID-19 is considered to be low based on multiple expert consultations, and it should not hinder vaccine development of conducting clinical trials, but that we do need to know that vaccine-enhanced disease relative to natural disease ascertainment is challenging, because specifically for COVID-19, there's such a broad spectrum of disease manifestations and pathogenesis that we continue to learn about, that we would want to make sure that we understand that before we can attribute vaccine-associated enhanced disease to any particular case. So we will need to understand as well that, as you see, the case definition does not have a level 1 of certainty, a definitive case, because that does not seem to be feasible at this time until we understand better the pathogenesis of enhanced disease, especially in the context of COVID. And so at this point, truly the main message is that we would recommend a systematic evaluation of all severe or atypical manifestations of COVID-19 disease that occur in vaccinated individuals to understand better and characterize better any probable cases or possible cases of vaccine-enhanced disease. Thank you.
Well, thank you, Floor, and apologies for all the technical glitches. Next time, maybe we do the webinar so it's so we don't hear people coming in and out. But thanks very much for this very clear presentation. I would suggest that we continue with Caitlin Dodd, and then afterwards we have the discussion. Thank you. Please, Caitlin.
Hello, and my name is Caitlin Dodd. I'm a pharmacoepidemiologist at the University Medical Center in Utrecht in the Netherlands. And I'm going to be talking a bit about the ACCESS project, which, as you can see here, stands for Vaccine COVID-19 Monitoring Readiness, and how we plan to identify COVID-19 disease in observational data sources. Okay, next slide, please. So the ACCESS project is a project funded by the European Medicines Agency with the overarching goal of readiness when COVID-19 vaccines are introduced. So in the ACCESS project, we will create harmonized event definitions, codes, and algorithms to identify adverse events of special interest. We're also creating common template protocols for calculation of background rates of adverse events of special interest. This protocol for background rates of AESI is the only one that will actually be implemented in the course of the project. The others are template protocols, which will be made available for researchers who would like to use them once COVID-19 vaccines are available. So these template protocols include those for safety assessment prospectively and retrospectively, both in EHR data and hospital data sources. Template protocols for effectiveness assessment using a test negative design and retrospectively using EHR data, and a template protocol for monitoring of coverage. We will assess feasibility to participate in studies. And as I said, we will generate background rates for adverse events of special interest using 10 data sources in 7 European countries. Okay, next slide, please. I won't go into detail on the adverse events of special interest we will be calculating background rates for. This list was based on work from SPEAC and also developed in collaboration with the European Medicines Agency. Go to the next slide, please. I'm going to go into some more detail on how we plan to identify COVID-19 disease and also levels of severity. Next slide, please. Okay, this is heavily based upon the work by Dr. Munoz and the Brighton Working Group. We have developed 2 proposed algorithms for identification of COVID-19 disease and COVID-19 disease severity in observational data sources. We've developed a broad algorithm, which includes 1 criteria for identification of COVID-19 disease using either the specific ICD-9 code or concepts to identify more broadly coronavirus. This is because we are using a number of data sources, all of which use different coding systems. So we've mapped these concepts using the Unified Medical Language System to different coding systems. Not all databases will have the availability of the specific ICD-10 code. The second criteria is presence of a code from one of the following concepts identified in the hospital settings. These include lower respiratory tract infection, pneumonia, bronchitis, indicators of respiratory failure and distress, hypoxemia, and mechanical ventilation or use of a ventilator. And third, death in any setting, and we have not yet identified the window following identification of COVID-19 disease in which to assess death, and that will be a discussion point. The narrow algorithm limits the first criteria to the specific ICD-10 code and will therefore obviously only be applicable in data sources using ICD-10 coding. Now, in order to define levels of severity, we've created the following criteria on the right. First level requires identification of COVID-19 disease without a record of hospitalization, and then proceeding through the levels, we require observations in the hospital setting for, as we proceed up the levels, lower respiratory infection, or lower respiratory symptoms, and hypoxemia. The next level, evidence of respiratory failure and distress. The fourth level requires evidence of mechanical ventilation or ventilator or dependence on a respirator, and level 5 requires the additional criteria of death. Next slide, please. The WHO working group has recently published a paper in the Lancet Infectious Diseases in which they describe a clinical progression scale that they suggest be used in clinical trials and prospective cohort studies of COVID-19 disease. Obviously, not every level of granularity will be possible to apply in observational data sources, but what we have done here is to map the levels of severity I described in the previous slide to this WHO clinical progression scale. So with level 1, obviously in observational data sources, we won't be able to detect asymptomatic cases unless subjects are tested and report their results back to their GP. However, with the level 1 criteria that I described before, we could pick up cases that are identified through testing, who are symptomatic, and then proceeding up through the levels, those who are seen in hospital, those who have evidence of respiratory distress, those who are ventilated, and finally, those who die. So we miss some granularity, but we can map the severity levels to this progression scale. Next slide, please. So as Dr. Munoz touched upon, what our algorithm identifies is actually a vaccine-associated enhanced respiratory disease because we focus on respiratory symptoms. However, other organ systems may be involved in severe disease, and here I've listed the other adverse events of special interest for which we have developed algorithms for use in observational data sources, and we could consider adding some of these events to our severity levels in order to identify vaccine-associated enhanced disease or severe disease in the absence of a vaccine in order to calculate background rates prior to vaccine release. So there's the option to include some of these events in order to identify other organ systems that may be involved in severe disease. And I believe we will now move on to discussion points. So I'll hand it back over to Miriam. Thank you.
Thank you very much, Caitlin and Flor again. I would like to give the word to Professor Stephen Black from SPEAC and also from the Global Vaccine Data Network, who will lead the discussion points. And there are several slides that actually presents questions. So first, there are 2 slides with questions that Caitlin, you created as discussion items, and we have received last evening a set of questions which have been forwarded by Vaccines Europe as a combined a question sheet. So some of them have been transferred into the slides. So I would suggest we go through these slides, and then Steve, I leave it to you to see from amongst the audience, either Floor or the Speak people or anybody else that could maybe address, or if people want to say something, that they make themselves known in the chat and then speak up.
Okay.
Let's see how we organize this. Yeah, thank you, Steve.
See how this works. Everyone, this is quite a big crowd for an informal discussion. I think one of the first discussion questions, if I may, is the one that was on that Caitlin just posed, is whether it's feasible really to look beyond vaccine-enhanced respiratory disease, because as we have learned with this virus, the manifestations occur really in every organ system, everything from appendicitis to myelitis to anything that involves either a vascular inflammation or organ inflammation. So that if you go beyond vaccine-enhanced respiratory disease, you begin to look at everything, which obviously you increase your chances of finding something by chance if you do that, and also it becomes operationally difficult to do. So I don't know whether Caitlin or Miriam would like to comment on the analytical difficulties there, and then we'll move on to the other discussion questions, or if others in the audience would want to comment. I can't see the chat, Miriam, so you'll have to let me know if people are.
Okay, good. So Caitlin, do you want to, because you posed the questions, do you have any idea about like inclusion of other organ systems in the seriousness classification? Because it's the question you pose. Do you want to give it a start, or what your thoughts are?
I can give it a start as a non-physician. I think it perhaps could relate to the first question here on the slide we're seeing, that that inclusion of other organ systems may be considered for development of algorithms of severity that are dependent upon other features of the population, for example, age. But that's just a starting point for the discussion, and I'll leave it to others.
Yeah, first of all, we need to remember that we don't know whether vaccine enhanced disease exists, and if it does exist, we don't know how it would manifest itself. Just because the virus was initially a respiratory virus doesn't mean the enhanced disease would be. So that would be an argument to look further. Are there people in the audience who would like to comment?
Hi, this is Surieta, FDA. I know this is a problem, and I'm not suggesting solutions are easy, but something that comes to mind is it will be easier to detect, to identify enhanced disease if we study that problem in populations for whom severe disease is not expected. Just to give you two examples, healthy first responders that will be vaccinated first in many populations, in many countries, and healthy children. For those two groups, the rates of severe complications are very low, under 1%, and in some cases under 0.1%. It's easier to detect an unusual increase in severe disease when we have a contemporary cohort of the risk to compare with. And maybe if the vaccine were even, if you can multiply that, this could be difference of 10 to 1 or even 100 to 1 that could be so extreme, so unusual that could at least create a signal, as Bob Wise did with rotavirus ages ago.
Okay, thank you, Hector. So maybe we'll move on to
Can
the
I
next.
say something, Steve? It's Barb.
Sure, Barb, go ahead.
Yeah, I was just going to respond to Caitlin's slide. I don't think autoimmune diseases should be included at all because we see them as one-offs in otherwise healthy people. I agree with Hector, but I was thinking more of like in the enhanced definition where you've got multi-system disease severity beyond what you would expect. Over.
Yeah, thank you, Barbara. That's Barbara Law from the Speak Project. Yeah, I think that brings us really to the slide that's up on the screen, which includes age. And we know that the manifestations of severe disease in children are quite quite different in terms of multi-system inflammatory syndrome. So that if you were looking for enhanced disease or disease in children, you would want to look for not respiratory disease per se, you would want to look for that manifestation, I would argue. And I think that goes along with what Hector just said. What about comorbidities? What do people, how would that affect the spectrum of enhanced disease? Do we know anything about that?
It's Barb again. I think it's now clear that the morbidities of diabetes and obesity and possibly older age, but are related to a increased inflammatory state, which could make the multisystem disease more likely. So I think we'd want to have a good idea of what that is in natural COVID disease.
Okay, so
this is Dr. Engler. Could I just ask a question?
Sure.
Yeah, so given that, you know, the autoimmunity, preclinical autoimmunity and preclinical cardiovascular disease are a huge factor in all our populations, and that gender differences, sex differences impact that as well. I think it's important to consider the vaccine hesitancy and distrust groups, that this is an issue that is important to people and not a small population. You know, in America alone, we have 40 million people who have autoimmunity. And the work of Dr. Judith James at Oklahoma Medical Research Foundation, particularly in relation to anthrax vaccine immune response variability in lupus patients and in family members of lupus patients, et cetera, I think might be to consider as you design or think this process.
Okay. What about, I mean, the next, thank you, Renata. I haven't heard your voice in a long time. There's also a mention of geographic and population specific variability. We've seen that the risk of disease is different in different parts of the world of symptomatic disease. And how do that, I think in terms of background rates, would clearly be a red flag that we need to be very careful about either racial or geographic variability in calculating background rates. The issue of influence of treatment or response to treatment, I think that is, there aren't that many treatments right now. We have dexamethasone and remdesivir, but there may be others soon. And that's going to potentially impact the severity score for these individuals, and would mean you'd have to control for that basically in your comparisons, I would think. Do others have comments on that before we move on to the next slide?
I just wanted to add that, you know, given the vitamin D data, you know, I think nutritional status may also contribute to differences in population and need to be considered as an important variable. And, you know, the suggestion of broad vitamin D supplementation, you know, has been published recently as well.
Okay, thank you. Maybe we go to the,
This is can I say something? This is Alena Kromova trying to say, can you hear me?
yes.
So I wanted to bring the notion that perhaps you know, the background incidence rate have to be looked within the clinical trial population where there will be a placebo group available. The points that have been raised is that we don't know, I mean, we assume the level of asymptomatic infection, but within the clinical trials, there is an active surveillance for both asymptomatic and symptomatic infections. I would like to say that maybe within the specific age group, you know, of course, the clinical trial population will differ from the general population, but the level, I would say, of severe disease, we should look within the same age group, and potentially, you know, the comorbidities were mentioned, but we know that diabetes, high higher BMI, living in the congregate settings are higher risk factors for the disease. And I would say we should not extrapolate the proportion of severe disease from one age group to another. And we should be careful also, not only the geographic representation, but even within the same metropolitan cities, we know that there are areas with higher transmission rate. So the other point that I would like to say that clinical trials ongoing may bring information also on the vaccine efficacy after one dose of vaccine received and by the serostatus. This is an information that will not be available, within the databases, because one of the points of definition is disease in the naive population. And unfortunately, within the healthcare databases, we will not know the prior exposure information. So these are a few points that I wanted to bring for the discussion.
Okay, thank you.
Hi, Steve. This is Hector. I have a comment that at least I believe it is important to consider. The first thing is, if we find an enhanced disease problem obvious enough, like a 10% increase, a 20% increase, a 30% increase, it doesn't matter what we do, we will find it. The real problem is on smaller rates of enhanced disease. And for that, we will really need to be much more strict. We will need a contemporary cohort of the same risk, either vaccinated with another vaccine or unvaccinated, to be able to tease that out. Otherwise, we will stay with the question, we will not be able to clearly respond to it. Background rates will not help us so much in cases such as those I am expressing right now, when the problem of enhanced disease is not too obvious. There are too many differences in infecting those: climate, nutrition, comorbidities, and even things we don't know.
Okay, thank you. We need to move on, though, I think. Next slide. So one of the issues that Miriam wanted to discuss is death, something we all think about occasionally, but it's complicated here. Is when do you identify it, and how long after COVID disease that you count cases? If someone's admitted with severe disease and dies, that's pretty straightforward. But what happens if they have COVID disease and then die 3 months later, 6 months later? Which of those cases would you include? And what about coding? If COVID is listed as a cause of death, do you count all of those, even though it may be 2 years later? How do you address this? Hector, you're an expert on death. Why don't you initiate the first comment?
The problem for me is that in studies we have been performing, individuals who are very frail and have a bad prognosis sometimes are sent to the hospice or even to home, and they simply die, sometimes even without being hospitalized. And we cannot ignore those cases. A way out of it, in a system in which at least cases are well identified, is death that occurs within a certain time after a diagnosis, a PCR diagnosis of COVID. Now, what that interval is, that's a problem, because the farther away you go from the diagnosis, the more likely you will have false positives. You can use 7 days, which is fairly safe, 2 weeks, which is still relatively safe, 21 days, and then you will have a higher rate of false positives, and 30 days, which is we have used in some influenza studies, which even, more contaminated, if you want. It's a decision to be made. If you want to be very specific and for investigation purposes, I would use a shorter time period after the diagnosis has been made. So something like 2 weeks, maybe even going to weeks.
Yeah, but maybe I can challenge that, because most of the deaths to COVID, if natural disease, occur at 3 weeks to 2 months following disease, because you have the initial infection and then you have hyperinflammatory syndrome, which occurs weeks later. So most of the deaths for COVID have occurred at week 3 or 4. So that would argue against using your shorter window, wouldn't it?
It is true in healthier populations. It's not true in the elderly and the frail, which are the ones who die in the largest numbers. So if you are working with a population of a healthier cohort, then you will have a more complicated situation and you will have to use larger. But for the very people, 80, 85, frail, that might not be necessary, at least not in the studies we have been performed.
Okay, thank you. So,
Steve, it's Barb. Can I just, the only point I wanted to make is to think about sudden unexpected death, because, and I'm probably biased because I read so many of the case reports that come out, but there are people who present as death or die in the emergency room from pulmonary embolus, stroke, etc., which could be a marker of endotheliitis and inflammation. So I just think to keep that in mind.
okay, thank you. So maybe I'm unfortunately going to have to leave in about 3 minutes for a WHO meeting. So I want to move on to the slide that says discussion items, vaccines Europe. And I think one of the key questions here is how, on an individual basis, a severe COVID case can be differentiated from vaccine-mediated enhanced disease. And this is, in a nutshell, what the Flor Munoz and her group have been debating for the last 3 months. And I think the conclusion is, and Flor, I'd like you to chime in here, please, as well, is that on an individual basis, in the absence of a biologic marker, which we currently don't have, you can have a high level of suspicion, but you can't ascertain whether an individual case is a case of severe disease and vaccine failure versus enhanced disease. Flor, could you comment?
Yes, thank you. And I have to say I appreciate the chat and the different points that have been shared in the chat, because it does bring up to this conclusion. I think that certainly if we consider that vaccine-associated enhanced disease occurs, it will potentially be a rare event. We do need to consider timing or temporality with the vaccine, and this is wonderful to hear about this specificity. Also, we need to think about we will not be able to identify this event likely. It's unlikely that we identify it in the context of clinical trials, that it will be once vaccine is given to large numbers of people. Yet those signals need to be very much looked at in the different categories of population that we have discussed, age, gender, comorbidities, all of these things. And the statistical portion of this will be clearly important. But I will say, although that would be the way to potentially identify this, the assessment of individual cases with some of those suggested laboratory evaluations does remain important, because that would be the only way that we would differentiate true vaccine-associated enhanced disease versus just severe disease or any other, manifestation of the disease.
Okay, thank you, Flor. So the next discussion item is if we can't differentiate between severe disease and VMED, the suggestion to consider monitoring the risk of VMED through monitoring vaccine effectiveness using severe or critical disease In other words, the proportion of individuals having severe disease who've been vaccinated before versus the proportion having severe disease who've not been vaccinated. I think that's a suggestion that I'm aware that the CDC is planning on doing in the United States. I don't know whether Caitlin or Miriam, if you'd like to comment and then maybe take over the discussion, because unfortunately I need to sign off.
Well, thank you very much, Steve, for guiding us till here. I think I don't have a ready answer to this question. And within the Access project, what we are doing is like the multiple items. So there is the background rates that Caitlin discussed, and then there will be protocols for more for studies, which could be effectiveness studies and safety studies. So vaccine-enhanced disease may actually, is part of the safety studies that are currently being written up in terms of protocols. For now, I think I would like to, because it's the hour, and I'm really grateful for the large crowd of people that joined. It does show that there is a lot of interest in this topic. The purpose of this meeting was to start sharing information and to start the discussion. We did not anticipate that we would actually find a consensus currently. I think we have achieved this quite well. I think the next step, certainly Vaccines Europe can come up with comments. Many people can come up with comments, and I think what we will need to see and address between SPEAC and VAC4EU is how we can bring this discussion further in an open and constructive and transparent manner. There are modalities to do that, either through webinars or to creating working groups or committees to do this. So I think for now, and I don't know, Bob, whether you want to say anything about this, but I think we will be announcing how we will carry this forward. So at least on the VAC4EU website, you can do this. The recording will be made available there. And I think we just need to brainstorm a bit on how we can get this crowd again together and solve this. Bob, over to you, if you're still there. Probably not. So then I would like to, what I would like to say is that stay tuned in on the VAC4EU website, on the page where you see COVID-19 vaccine monitoring. We will be announcing how we bring this forward. So thank you very much for your presence and participation, and especially the speakers for the presentations that they held. Thank you.